I would love to introduce our first speaker. We have Keba Lebani, and Keba, you may wish to go online and put your camera on. So I'm delighted to introduce Keba. She is a biomedical scientist and biotechnologist. As a lecturer and researcher in the Department of Biological Sciences and Biological at the Botswana International University, BIUST, since 2015. She joined BIUST after her academic research PhD studies here in Australia in Brisbane, where she worked and assessed the antibody engineering for diagnostic applications. During her doctoral studies, Keba was Staff Development Fellow, being developed as a future academic staff for the Botswana International University. And she was also an awardee of the Schlumberger Foundation Faculty for a Future Fellowship. So great achievements for Keba at that stage already. Her current work spans a variety of disciplines from the biomedical sciences to epidemiological surveillance, diagnostics development to commercialization of fermented products. Currently also supervising a lot of students in the aforementioned fields, and she's established key collaborations to drive the research. She's a keen advocate for STEM, women in science. So Keba coordinates the mentorship for girls excelling in maths and science, GEMS, and also internationally mentors female postgraduate students. Keba leverages her professional network to really support all the aspirations of her mentees. So with that, Keba, I would love to open the floor to you, where you will be presenting today on a key topic that is, oh, sorry, no, my slides are gone. Where is... And I can't even find my slide for the title for your presentation, Keba. That is, can you help me, please?
Thank you, Sandra. So I will I will just be talking to the audience about being resourceful in an under-resourced research landscape and talking about how we can leverage optimism and collaboration to navigate those challenges.
Thank you.
Yeah.
I'll leave the floor.
May I go ahead and share?
Yes.
My slides.
If you just go and present and view.
Are we all good?
Yes.
Okay. So a very good day to everyone joining today from across different time zones. I'd like to take an opportunity to especially thank Sandra and the Next Gen VAC group for inviting me to speak today and shining a spotlight on Southern Africa today. I see Sandra. Judie will be coming on next from South Africa. So as already introduced, my name is Lebani, but everyone calls me Keba for short. So I'm going to be talking about how you can be resourceful in an under-resourced research landscape. So my presentation will not necessarily be highly technical, but we'll touch on some scientific aspects. So I think I would like to preface my presentation by maybe addressing why perhaps some people might be surprised for someone from Botswana to talk about under-resourcing, particularly because Botswana has been branded worldwide as the Diamond Nation. But just to highlight that resources vary across different landscapes. And according to the World Bank rankings, Botswana is classified as an upper middle income economy. And this poses some problems for us in the research space because it limits the amount of funding that we are able to seek out. We are not classified as a low or middle income country. So it means that we have to really fight for competitive grants with advanced economies. So this challenge is also compounded by the fact that the government of the Republic of Botswana does not especially earmark funds within their budgets for research. So we do not have a national research fund. We also in Botswana don't have a real postdoc system. So most people in the audience may be aware of the postdoc system where new doctoral graduates get an opportunity to really fine-tune their research ideas under the mentorship of I guess, experienced staff. Another issue which is compounding the research landscape in Botswana is that capital investment for research is mainly done in the form of equipment. And this is sort of done in an ad hoc manner without real intentionality about also earmarking funds for running that equipment and maintaining that equipment. So all of those things make being a researcher in Botswana very tricky business. So today I'm going to be talking to you about how we can leverage optimism and collaboration to navigate that space, hoping that this will be something that may be useful to people that may find themselves in a similar opportunity, in a similar situation. So basically that is the outline of my talk. I'm not going to go too much into the science. As I said, I want to talk about what the resources that are lacking are and how can we be resourceful in trying to navigate that by working with others and also having a giant scoop of optimism on this side. So I've already been introduced by Sandra. I started my training in biomedical sciences. So all of my tertiary education was in Australia. I did a Bachelor of Biomedical Science at Monash University, and then did both my master's and my PhD at the University of Queensland in Australia, which is where I met Sandra. What do I do now? I am a lecturer teaching diverse modules, one of which is in applied immunology. And I'm also a researcher working across different research foci that I'll talk to you about. And also importantly, which is something that I do like to talk about, is some of the work that I do in service as a steward for STEM. And also for gender empowerment, and really trying to make sure that everything is done with intentionality and real, I guess, sincere efforts towards empowerment and not it being about tokenism or optics. So I'm pictured there. This is me in the middle many years ago when I was still a fresh-faced employee of the university. I think this was 2018, with some students that I was mentoring from a secondary school within our locality here at Bust. So my research work, as I said, is quite broad. When you find yourself in a situation where you have just finished your PhD and you have to come and work within a university and sort of stand on your own without real mentorship, you have to learn to be a little bit clever and resourceful and figure out where you can fit yourself until you hone in on what is going to be your research focus. So I work across different fields. In the biomedical science space, we're looking at HIV and hepatitis B virus separately and together as co-infections that are prevalent in the Botswana setting, and looking at how those viruses evolve differently in response to treatment and the subsequent, how would I say this? So the subsequent results that could arise in terms of drug resistance. Also working in, so the work that I do in HIV and hepatitis B virus, I'm doing with the wonderful people at the Botswana Harvard Health Partnership. Also working with a group from the University of Sydney on host genetics and prostate cancer. So looking at different groups of people with prostate cancer. So we're at the moment looking at Caucasians versus Southern African cohort to see if there are differences within their germline and their somatic cells that are resulting in differences in how prostate cancer manifests itself. So we're looking at whether there are new biomarkers there to be explored in the space of diagnostics and also as targets for treatment. I'm working with a colleague, Dr. Nerve Jo, on traditional fermented foods and beverages. This one is sort of my hobby research topic. It's just something that I'm interested in. And I've been learning a lot in that space from Dr. Jo, where we're trying to isolate novel microbes from the Botswana setting that have competitive commercial fermentative traits that can be useful in product development. I also do some, sorry, I seem to have scrolled my slides. I do some work with another group of people, some lovely people from NADI, which is our National Agricultural Research and Development Institute, where we are looking at ticks and tick-borne diseases. So tick-borne diseases are diseases of economic importance. The agricultural sector in Botswana relies quite heavily on livestock and livestock products, and tick-borne diseases can really undo some of the good work that is being done towards food security and also generating revenue for the economy. So we're just looking at making sure that we have records of the diversity of ticks that are prevalent across different areas in Botswana and the pathogens that are vectored by those ticks. And I'm also working with them on trying to improve diagnoses of different diseases that are vectored by ticks. And within the management space, one of the key ways that you would manage ticks is through the use of acaricides. So what we want to make sure is that we are limiting establishment of acaricide resistance within the livestock sector. So my focus today is going to be on the work that I do on foot and mouth disease virus. So this is work that I do with some wonderful people at the Botswana Vaccine Institute. And I've also been fortunate enough to do some work as well with the vesicular disease unit at the World Reference Laboratory for FMD, which is hosted at the Pirbright Institute in the UK. So within foot and mouth disease, I'm primarily working on diagnosis of the disease as well as characterization of the virus, but we're trying to move towards vaccine development. So just in case you thought that I'm an imposter and I'm not going to talk about vaccines today, we'll get to that space, but I'm hoping that Judie will talk more towards the technical aspects of vaccines and vaccine development. I'm going to just be presenting my story about doing research in an under-resourced landscape. So we'll talk a little bit about some technical aspects of the disease and the virus so that you can understand the work that we're doing in this regard. So foot and mouth disease, also abbreviated to FMD, is a transboundary veterinary disease of economic importance. Transboundary, I guess literally just meaning able to transcend boundaries. It spreads rapidly and easily. It affects cloven-hooved animals, so this is not a zoonotic disease. But as I've already emphasized, the livestock sector is very important to the economy of Botswana. So transboundary diseases, generally because of their ease of spread, they require international cooperation to control them, as well as to coordinate responses to the disease. So when you have outbreaks of FMD, they affect countries that are reliant on animal and animal products, because then you are not able to export any of your animals or your products. So if you are reliant on agriculture, this can adversely affect your economy. So the disease is caused by picornavirus, which is called foot and mouth disease virus, which is abbreviated to FMDV. And the virus is non-enveloped, and it has about an 8.2 to 8.4 kb RNA genome. And this virus, I guess, its genome sort of similar to, for those that are working in the human health space, similar to hepatitis A, and also to polio. So this RNA genome depends on an RNA-dependent RNA polymerase for its transcription. And this polymerase is error-prone, and it does not have proofreading mechanism. So as it replicates, you do not get that proofreading. So you get an accumulation of mutations within the genome. So you get a lot of variation within the virus. The virus is vectored. Well, I guess it has a reservoir in buffaloes within the African setting and also in Asia. So there I'm just showing you the genome. When it is translated, it yields this polyprotein that then gets co- and post-translationally cleaved into 4 structural proteins, which are your VP1, 2, 3, and 4. VP1 is especially important for characterization of the virus and also for genetic and antigenic drift. And then you get you get 8 non-structural proteins. So the virus has different serotypes, 7 serotypes, 6 of them are circulating, and they are AO Asia 1 in the Southern African territories, serotypes SAT1, 2, and 3. And these different serotypes have different geographic distributions. And within those serotypes, there's further variation that then gets the viruses classified into different topotypes, which are geographically contained in different lineages as well. So these geographic distributions also lend themselves into a different classification system, which we call pools. It just means that in different regions, there are different combinations of viruses that exist. Within our pool in the Southern African region, we're in pool 6, and what we predominantly have circulating are SAT1, 2, and 3. So importantly, The different serotypes result in clinically similar disease. They have some slightly different, so they have some slight differences in their virulence and also some of their transmission pathways and also their stability. Their thermal stability differs a little bit, but the disease that manifests is the same. But what is different is the immunological responses. The immunological responses to each of the serotypes is not cross-protective. So you need to think about how you formulate vaccines to make sure that your vaccines are going to cover the virus that circulates within your region. And I hope that this already sort of highlights a potential issue in the control of FMD, which is why it is important that there's coordinated responses to outbreaks, because if you get what you call an incursionary virus, so one that does not typically circulate within that region, it means that the livestock within that region will not have any potentially neutralizing antibodies to react to that virus. So the primary control of the disease is through vaccination. And as I've highlighted, the formulation is important to make sure that you get appropriate immunological responses to the vaccine that is being administered. So, there are some questions to ask ourselves around FMD. And when there is an outbreak, what you need to know is that you need to know whether what is causing the outbreak is FMDV. Clinically, you would be looking for blisters in the mouth and in the feet of those animals, as the name says, foot and mouth disease. But these symptoms can arise from other infections. So how we find out whether it is FMD, you would have to propagate the virus in permissive cells to see if it results in a lytic response where we see what we call a cytopathic effect. So you also need to know what the serotype is, because as I've already said, there's no cross protection. So the work that I do there is in finding out how can we better diagnose FMD, and our research efforts are in development of cell lines to make sure that we are able to isolate the virus fast, and also in development of serotyping molecular techniques to make sure that we're able to say that it is this particular serotype as quickly as possible. The lack of cross protection also means that it's important to ask yourself whether the field strain and the vaccine strain share the same antigenic properties to ensure immunological cross protection. So as I've already said, that RNA-dependent RNA polymerase is error-prone and doesn't have proofreading ability. So even within serotypes, you start to get some changes or accumulation of mutations that can take away that immunological protection within a serotype. So you need to be able to check if your vaccine is going to cover the virus that is within the field. So what you would have available is serum from animals that have already been vaccinated with your vaccines. And you would want to incubate that serum together with the virus from the field. And then you would add those to infection permissive cell line to see if there is any residual virus infectivity that was not neutralized by the antibodies within the serum, to ensure that your vaccine will actually cover that strain, because if they don't, then you need to look to an alternative vaccine strain for the outbreak that is going on. So we do a little bit of work in trying to ascertain which are the best in vitro assays for determining antigenic drift and vaccine escape. And also the origin of the virus is important. The concerted efforts in managing foot and mouth disease need you to know where you need to strengthen control measures. So knowing the origin of the virus is not really a blame game, but in order to help each other to strengthen protective efforts so that you don't get further transmission. So in terms of doing some fine mapping, I've also been working with the group at the Fulbright Institute on the pipelines for whole genome sequencing. So the research challenges, the under-resource, the lacking resources are in terms of research space. Foot and mouth disease virus, it requires high containment facilities, BSL-3. There's only one facility in the country, and it is obviously prioritized for routine work in diagnosis and surveillance. So we don't get a lot of time as we would wish to work on research questions. Because the space is limited, you also get a limited number of students. You then don't have the opportunity to, I guess, pursue different avenues of answering a research question that we have. There's also limited research scope. As I said, I'm working with the Botswana Vaccine Institute on this work, and they're a business entity, so they have to put forward some of their business interests. We cannot necessarily focus on just primarily academic questions. In the absence of working with others, it means that you have limited study samples. Fortunately, Botswana hosts a reference laboratory for the Southern African region. So we do get a bit more samples than others would, but it would be better to have samples from elsewhere, which is why I've gone out of my way to ensure that I also have linkages with the World Reference Laboratory. There's limited funding. When you partner with a business entity, you're not always able to access research funds. So there are some challenges there in trying to navigate that space, but also limited equipment. The business entity buys what it needs in order for it to perform its function. I might be interested to find out perhaps what the differences in the immune responses or the cellular responses to SAT2 versus SAT3. And I might want to have a flow cytometer there. That is not their interest. So, you know, you have to figure out how you're going to navigate some of those challenges. So the solution to most of these challenges, as I've already said, for me, it has been in collaboration, making sure that I get invitations to join industry projects and being able to apply for funding jointly with other organizations that meet the requirements of funders, things such as research visits, going to learn a particular skill. As I've already said, you know, you have to work with what you have. If I want to learn a particular skill, but I'm not able to access a biosafety level 3 lab, then I might be able to work on hepatitis A, which is similar in its makeup with FMDV, but I can work with it at a lower biosafety containment level. Research fellowships, sabbaticals, finding opportunities to have adjunct or associate positions. These are all important ways in which you can collaborate with others, and finding yourself there requires you really to network as best as you can. You know, go to the think tank meetings, go to the conferences. You know, I think the think tank meetings, we call them pizzos in Botswana. I think in South Africa they call them indabas. So these are places where you can talk to people, but it is not highly formal, and you can bounce some ideas around, and your collaborators are the people that are going to put you on to the next collaboration. So why both optimism and collaboration? I realize that I'm running out of time. So collaboration is a relationship like any other, and collaborations are inherently complex. So you really need to have optimism because you have to believe in the shared goals of the collaboration. You also have to know that there'll be diverse perspectives. If I suggest something to my collaborator and they go with another idea, it shouldn't be within me to hope that that idea that they go with goes horribly wrong. I should put my best foot forward to try to make that idea work as best as possible. But you also need a whole heap of optimism, because in research, there are always going to be setbacks. It's not a linear process. So patience and resilience heavily depends on optimism. And collaboration really is a belief in people, and to believe in people, you need that optimism. So my next steps within FMD is trying to work on novel ideas to develop next generation vaccines for FMD. Currently, what is being used are inactivated viruses, which really have a limited immunological response, which is dominated by antibodies, and you don't get a lot of your cellular responses to the vaccine. So there's a requirement for boosters. You know, agriculture is about numbers, or livestock farming is about numbers. You need as many animals being productive. But if you have high numbers and you need to vaccinate for FMD, and you need to do it 3 times in a year, if you are in an area that gets FMD outbreaks, then this is expensive. So we're trying to come up with ideas of what we can do to improve the immunological response, and also what we can do to not only necessarily just look at region-specific formulation, but towards a universal vaccine for FMD in order to cater for any exotic introduction of the virus. So what we need, the collaborative inputs, obviously funding, have some leads with technical expertise, and the hands that need to do the work. And as I've already said, a huge scoop of optimism. I'm going to end there, and would like to acknowledge some people from the Botswana Vaccine Institute. Their names are there. Ms. Tepang Kabelo at the University of Reading, Manamwidi Gesamang at B-Tree, my current MSc student, Petronella Msholombe, and the people that I work with at the Pirbright Institute, Dr. Andrew Shaw, and the vesicular disease team. Thank you, Sandra.
Thank you so much, Keba. That is definitely an interesting talk, and it really highlights also the challenges that you're facing there. That is very different to many of the developed countries where you just go and order something and you don't have to look for samples, so it's much easier to access them. And with your permission, Keba, in the interest of time, I would love to welcome Judie, and we take all the questions at the end of that, if that's okay.
That sounds good. Thank you.
Thank you for your talk. Keba. Judie, with that, I would love to welcome you on and do a quick introduction for you. So Judie, you're currently a postdoctoral research fellow at the Africa Institute Research Institute, AHRI, in South Africa, and a global health alumni of the Harvard Public School of Health. At your current institute, you are committed to improving health outcomes in under-resourced communities, with the research at the forefront of advancing the HIV immunotherapies and innovative vaccine strategies. Jodie leads the studies on the B-cell repertoire characteristics in South Africa, which is crucial for designing germline-targeting immunogens capable of eliciting a broadly neutralizing antibodies. This work was highly significant for the development of the investigational new vaccines. Judie's research also focused on the potential vaccine effects in the host following broadly neutralizing antibody infusion, offering vital insights into future interventions. Through the collaborative science, Judie has dedicated to generating insights that improve the immunological outcomes and strengthen health systems in settings where they're needed the most. So with that, Judie, I would love to welcome you, and your presentation today is addressing the mapping the B-cell precursors in HIV, a Southern African perspective on germline-targeting vaccine strategies. Welcome, Judie.
Thank you, Sandra, for that introduction. Please do confirm that you can hear me.
Yes, I can hear you loud and well. And if you can share your screens, your slides, please, then we're good to go.
Okay, I am just finding them. Okay. While this is happening, thank you, Sandra, and thank you to the organizers for inviting me to share my work.
Thank you. We can see the slides now.
Okay, everything is all good. All right, so my talk is actually very technical, very sciencey, with all the sciencey jargon, very different from Keba. So what I wanted to do here is to just highlight some of the work that we are doing in the vaccine space, HIV vaccine space, in an African context. And the title of my talk today is Mapping B-cell Precursors in HIV, a Southern African Perspective on Germline-Targeting Vaccine Strategies. So the first thing that I want to highlight is why we still need HIV vaccines. I think the short answer to that is 40 years in, we still have no cure for HIV. We do not have a licensed HIV vaccine. And coupling that is that as of 2023, we have nearly 40 million people living with HIV, and we have 1.3 million of new infections every year. So these are indeed staggering figures, right? But having said that, we also know that we have made great progress with ARTS, which has shown to be effective. Okay, just trying to move this thing that's in my face, but I can't. All right. So ART has been effective in suppressing the virus, but we still have challenges with its lifelong use. We also have cost challenges with the newer modalities like the long-acting injectables. There's also great concern with the shifting HIV funding status, which could potentially result in derailing or setting us back on all this progress that we have made. So because of that, the field is still lobbying for HIV vaccines, because traditionally we know that a vaccine is able to provide cost-effective, broad, and durable protection. Right. So there are so many strategies that are being tested in the clinical setting. And one of those strategies that is most advanced is the germline targeting approach. And this is currently offering 3 candidates in the field, which is your EODGT8, BG505, so CPT 1.1, as well as the 426 core, right? So what is the germline targeting approach? So this approach comes from the understanding that a few individuals that are HIV or chronically ill are able to produce broadly neutralizing antibodies. Now, using advanced techniques in the lab, researchers have been able to revert back to say, where did this broadly neutralizing antibody or where did this BNAb come from, right? So they have been able to refer back to where it started from. So we now know what the starting B cell looked like. We now know what its signature features are. And the idea is to now to bring in vaccine candidates that are engineered in the lab that can activate or prime the starting B cell. These are class specific. Once it is primed, we come in with a sequential of other immunizations or immunogens that have got increasing complexity that are able to shepherd that starting B cell into producing broadly neutralizing antibodies. So this is a series of immunization, which we understand in HIV is the way things are going because you are not able to do it in one short immunization, right? So for our study, we are focusing on the starting B cell. Okay, so the questions that we are asking regarding the starting B cell, right? We want to understand that in the pool of the B cells in our cohorts, we want to characterize them. We want to be able to define, do they have potential of becoming broadly neutralizing antibodies? Do they already bear the signatures or the features that we are looking for in broadly neutralizing antibodies? We also want to assess the impact of external factors. In our case, we are looking at the impact of HIV disease. We are also looking at the time when AHRI is initiated. The last thing that we wanted to look at is to evaluate class-specific precursors, right? As I mentioned, there are already HIV candidates that are in the clinical setting. So we want to have an idea if we have the specific starting B cells that will be able to produce that kind or that class of broadly neutralizing antibodies. So to answer these questions, we are using the FRESH cohort. This is an established cohort in Umlazi, Durban. It is a cohort of female participants that are aged between 18 to 23 that are at high risk of HIV acquisition. They are followed for a very long time and they are tested for HIV twice every week. And because this is a longitudinal study, we have the privilege of having stores and stores of archive samples because the sampling goes on for years and years, right? So for our study, we combined M1 and M2. The idea is we wanted to define the overall B cell repertoire and we wanted to evaluate the impact of HIV. So to do that, we used bulk RNA sequencing. So this is cheaper on cost. So because it is cheaper on cost, we are able to do the longitudinal sampling, right? So for this study, we had individuals that started off as HIV negative. Some of them became people living with HIV, and then they were put on AHRI treatment early within 1 week of infection. In the same group, we have other individuals that acquired the HIV disease, but they were only put on treatment later, at least 1 year post-infection. This is because we still have, remember, this cohort is sampled for a very long time. So we have a group of these individuals that were put on treatment late because the guidelines hadn't changed. Then you didn't have to put on treatment upon detection. We also have another group that is just before the late treated. This is the chronically ill individuals before they go on AHRI. So the advantage of doing the bulk RNA sequencing, it allows us to do this at low cost. It also allows us to have some depth. It allows us to include this longitudinal sample. The disadvantage is that we will not have, we will end up with unpaired chains. We don't have the pairing of the heavy and the light chain. So we are not able to define the specificity of the BNAb or the antibody. Right, so what have we found so far, right? So we understand that an antibody is defined by its V, D, and J usage. It is also defined by its CDR3 length, both in the light chain and the heavy chain. For some cases, there is signature motifs that you could find on specific position of the gene. So our first question was just looking at this pool of B cells, what are the overall characteristics? What is it that we are seeing? So on the VDJ gene usage, the ones that were most prominent was the VH3 and the VH4, the D3, as well as the J4. So when we zoomed into the VH family, which was the most used in our cohort, we found that the most abundant gene of use was the VH330. Then we went on to look at the CDR H3 because this is also used to define the antibody. For our cohort, the median was between 14 to 15 amino acids, right? So all this is similar to what has been reported before. But what was of interest to us, we wanted to be able to inform the newer vaccine candidates that are looking at B cells that bear a long CDR3 length so that they can target the V2 apex, right? With the understanding that most of the immunogens or the candidates that are out there are targeting the CD4 binding site. So now there is also a move to say, what of other epitopes, right? Instead of just focusing on the CD4 binding site, let's look at other epitopes. So another epitope of interest is the V2 apex, and it requires a long CDRH3 length. So this is what we wanted to explore to see whether in our cohort we do have those. So when we looked at the naive repertoires, right, which is your IgM, we didn't see much difference. But then when we looked at the IgG, which is the memory, we do see expansion in the pre-art. We also do see expansion in the early treated group. So this suggests that disease exposure is probably expanding the length, okay? Then we went on to look at HIV-driven changes in the B-cell repertoires because we wanted to know what the impact of HIV is on the repertoires. We looked at the somatic hypermutation rates, and this was as expected. We didn't see any changes in the naive repertoire, but we do see expansion in the IgG, which is the memory, and this is as expected. But what was interesting for us is that when we looked at the individual or specific genes, when we're just trying to see what happens at gene level, here we also wanted to see the impact like relative to the time of art. So we looked at the late treated group and the early treated group. We were looking at what happens before they are put on art and then what happens post-art. So in the late treated group, you can see there is sort of a depletion, a drop, which then seems to recover in some instances and in others it doesn't. But in the early treated group, there is almost a similar trend for each gene where the abundance or the frequency or the proportion of the gene of interest is the same pre-infection and even when they are on art. So we are suggesting, this probably suggests that the time of art could play a role in maintaining these profiles in HIV. Then from there, we moved on to AIM-3. So the AIM-3 for this study was to evaluate class-specific precursors because we wanted to inform class-specific broadly neutralizing antibodies, which is the VRC01, informing also the use of the EODGT8, which is an immunogen candidate that is already in the clinical setting. So for this study, right, we didn't go with the longitudinal angle because this requires us to start with a big amount of the PBMCs because the cells of interest that we are looking for They're specific, they are rare, so they come in very, very small amounts. So for us to be able to do this, we needed at least 200 million starting PBMCs. So because of that, we ended up only selecting a few donors from each group. And the average time on AHRI for the early treated group was at least 1.5 years. And then for the late treated group, this was about 2.5 years. For this, we use fact sorting, right, flow cytometry, and then we use the EODGT8 as the bait so that it can fish out the cells that we want to analyze. We also use total seq because the numbers are very small. We want to be able to pull as many donors as possible. This here just shows our getting scheme. We were sorting for total B cells and then just the ones that bind to the EODGT8 bait. From there, we then create our libraries using 10x Genomics. We are using the Illumina NextSeq platform for sequencing, and then we use the available post sequencing tools that are available on the 10x Genomics website. Here, we're just showing you the outlook or what we see from the BDJ Loop browser. Okay, so what are the results that we are currently looking at, right? So here we were looking for a specific signature. We wanted to inform whether we have the precursors to a specific class of broadly neutralizing antibody, which is the VRSCO class. So the first thing that we looked at is, do we have the gene that is specific to that class? So here we just analyzed across all groups, which is the early treated, late treated, as well as HIV negative. So yes, we did have the correct gene being used. We also identified other gene usage to non-sensitive HIV sensitive broadly neutralizing antibodies. So once we identified the heavy chain, we also identified the light chain, and we had the good pairing that we were looking for. Then the next thing is then we looked at the signature, which is a 5-amino acid light chain, right? The CDRA length of the light chain is 5 amino acids. So that's the second feature or the third feature that we're looking at in those sequences, whether our sequences bear that signature feature. We only found that in the early treated. We didn't find it in the HIV negative or the late treated group. So the question that we think we should be asking next year is, does early treatment maintain any naturally primed VRSCO1 class specific features, right? Why do we only see it in early and not in the other groups? And then after that, we then focused on this early treated group because we wanted to see whether we have all the other features that we were looking for. So yes, we ticked the box for the VH, for the heavy chain that was used, and it does, from those that did use that gene, they had the 5 amino acid length of interest. Then we went on to look at the logo sequence, because for this group of BNAbs, it has got a signature motif that we were looking for. So that signature motif, you can see that in the mature BNAb, we are looking for the QQY right XX. So in our early treated group, we do have that enrichment of the signature motif, probably meaning that this is already primed. It's already going in the right direction, which could speak to strategies that are trying to introduce immunization in people that are living with HIV already. So we think this could lead to knowing that these people already have some priming that has happened because of the disease exposure. So if you bring in an immunogen, you're probably starting at a little bit advanced level than you would in people that are not exposed to the disease. So to conclude, we were able to inform germline-targeting vaccines implication in the sense that from the bulk sequencing, though it is a general overlook with no specificity, we do identify which genes are being used mostly. So from there, you'll be able to say, okay, maybe immunogens that target classes like the CAP256, which uses this gene, will probably be more feasible in this cohort in comparison to others. That's the kind of questions or answers we want to provide. We also saw that in the early treated, there could be some preservation of the B-cell repertoire profiles. Then when we moved to the antigen-specific, this same trend is noted where in the early treated group, there was some preservation of the FURAC1 class potential. So all in all, we think vaccines that target the CD4 binding site, potentially the ones that also target the V2 apex, could be feasible for this cohort. Currently, we are now looking at functional validation of those B cells that we were able to select out or fish out from the pools. So with that, I would like to acknowledge the fresh participants, our donors, and everyone who has made this work possible. Thank you, Sandra. Back to you.
Thank you so much, Judie. That's really great and impressive how much work you have actually put into that, because it looks like a lot of lab work and a lot of studies and a lot of time. Lots of people involved in there.
Yeah,
Keba,
thank you.
so thank you very much for that. Keba, if you also want to come back online with the camera and microphone on, there are some questions online. I'm just checking right now. So I had two questions. So Keba, I'll just start firstly with you. How easy or how difficult is it for you to compete with high-income countries for the grant attribution? And do you have any suggestions for other young LMIC researchers with regarding the funds? And same for you, Judy, you can answer after that as well.
It's not easy to compete. It's not easy to compete, but I've had to take a big spoon of patience with this, because I think when you're competing with experienced researchers in developed countries who have a track record of having managed funds and also having high-level output, whereas looking at your local setting where you might sort of be doing baseline studies as opposed to that translational high impact research, it becomes really difficult to compete for funding. But I guess what I was trying to get at is that collaborating with people that have that experience, then they can include you in some of their research proposals, and over time, you are a co-investigator in their work, and you start working towards some of your own independent output. So it needs a lot of effort. Otherwise, you'll get frustrated and throw in the towel.
Is that the same experience for you, Judy, for your work?
I think so. I think Keba does really paint the picture. Yeah.
Judy, a quick question for you. So in particular, have you also looked into some of the difference between the states and different cohorts or different outbreaks when you have the HIV regarding the epitopes? Are there any differences?
Okay, so I think to answer the first question, whether we have looked at other populations, right?
Mm-hmm.
Okay, so this is the idea that we were doing this in collaboration with some individuals in Kenya. It's still ongoing. So we wanted to do the epitope specific for the CD4 binding site in the South African cohort. And then our collaborators in Kenya were looking for the same in the Kenyan cohort, and then would compare the two. We are not there yet, but we are heading towards that.
Mm-hmm.
The second question?
Oh, it's just if you had already started to look at that, if you saw a difference in the repertoires, but if you haven't started to look at that yet, then it's maybe yet to come.
Yeah.
Yeah. And then the other question is also for you, Judie. Regarding the feasibility for a treatment, the clinical trials, how easy for you is it to, when you propose, for instance, whether CD4 binding sites could be a potential target, how practical, feasible is it to actually test that in the subjects, so in the actual patients?
Okay, that's a great question. So we know that the IRV is in the forefront of this. They're already doing this. So they've done it through the G001, which was a proof of concept, and they introduced the EOD-GT8. They wanted to see if it does prime the correct B cell. They've now done the G00 trial, which looked at participants in Rwanda and in South Africa here. So they've already also tested that in the clinical trial. So there are things that are already happening in the clinical space. So this study was also funded by IRV. So it's just feeding into that information. And if we want to bring it into the clinical setting, IRV is already doing that. So it means it just flows through that.
No, excellent. Now I'm also mindful of the time and all the attendees that are staying past the normal time. Can I ask one last question to both of you in a few words? What are your suggestions on efficient networking, knowing particularly the challenges that LMICs have to participate in congresses, in conferences where it's hard to get funding or even to travel out of Botswana? For Keba, any few words out of advice, please?
Well, could you kindly repeat that for me, Sandra?
What about, in a few words, the suggestions on efficient networking, in particular knowing how difficult it can be for LMICs to really participate and go to congresses, conferences that are expensive, or you might even be limited to leave your country, right? Any few words of advice there?
So yes, so that is something that is challenging, particularly with the research, with the financial constraints going on at the moment. But as I've said, even attending meetings at the local scene is important for you to get to meet the people that are making important decisions regarding whatever problem you're working on. But I think a positive thing that came out of COVID is that more so you tend to get hybrid conferences that give you an opportunity to attend virtually. It's not always best to do so. You know, you are not there at the dinner able to just have a conversation with someone. But the webinars allow you to listen to people, and even when you send the cold email, you say to someone, I listened to you on this platform talking about this, before you then sort of propose something that you could work on together. So it helps in some regard.
Thank you.
I think that is true. For me, I was going to say that platforms like this, where you invite us to come and tell our stories about our work, it exposes us, because people now know that I am doing this. I don't necessarily have to be at a conference, right, physically. But webinars like this, they do showcase the work. And I tell you that almost all the time after a webinar, you will have more LinkedIn requests for connections, because people now know you. So that's how it goes. Then you're able to connect through LinkedIn. They're able to follow your work, and
no, that's excellent.
Yeah.
Now, I did see there was one question from Flyball Kawonga for Keba regarding vaccinate for FMD after an outbreak that occurred already or during. Won't this be catastrophic, having an effect on the animals? If you don't mind to answer the last question live, and then everyone else can answer on question through LinkedIn.
Yeah, so you do vaccinate, and I know that this is not a common strategy for control, because you don't have any specific treatment for foot and mouth disease, you know, other than supportive strategies of making sure the animals are hydrated and everything, because when they have blisters and such, they cannot eat well, but at least hydration is something that is important to try to minimize morbidity in those animals. So vaccination is just meant to strengthen the immune response within the population, and also to ensure that you do not get further spread just in case there are some animals that are not covered by vaccination, you want to make sure that you put that up. But within those that may be early in infection, having that boost of the immunogen is helpful for the immune response. So it's a very expensive and labor-intensive undertaking to control foot and mouth disease.
Awesome. Thank you so much. So with that, I would love to conclude this webinar session. Thank you so much for our speakers, Keba Lebani and Judie Magura. It was a pleasure to have you on board today. And we look forward to the next webinar from the next vaccinologist. Thank you, everyone, and good evening. Goodbye.
Thank you so much.
Bye.
Bye.